I think all of you are familiar with the tree of life. So you can see that the different groups of the different species there, they are uh, grouped according to their certain similar features. Here, here, this also we are also in the different era, how the different, like for example, the dinosaurs, how they became extinct, and only some of the descendants like the birds and some of the reptiles are still alive. So today, as I said, so we talk about the lectures on the phylogenetics. So what we go to different terms of the phylogenetics, then the different algorithms. So you don't have to worry about the algorithms. As I said, if you're a molecular biologist, if you can, the next lecture we will do the hands-on on how we can do the phylogenetic analysis using the different softwares, especially the Mega and the Fig, how you can edit your trees using the Fig tree. So today we'll mostly cover the theoretical aspects. So we know the differences between the taxonomy and the phylogeny. So taxonomy is the science of how we classify the organisms. So there is a basic difference between the taxonomy and the phylogeny. Phylogeny actually, so you try to find the evolution of genetically related group of organisms, or we study the relationship between the collection of things. For example, we can look into how the genes are related, how the proteins are related, how the different organs are related, also some phenotypic characteristics as well. So why we didn't need to carry out the phylogenetic analysis? So there are for several reasons, and one of the things is like to find the evolutionary relationship between the organisms and find the relationship between an ancestral sequence and its descendants, as I said. So, for example, we have the ancestor and how they relate, how the descendants are related to each other, and estimate the time divergence between a group of organisms that share a common ancestor. So for example, how many million years ago, millions of years ago, or how many years ago, so one sequence diverges from the sequences diverge from the ancestors. So that's what we look into using the phylogenetic tree. So basic problem, as I said, that we look into that we infer the complete ancestry of a set of objects. So objects, these are the objects which we can observe at this moment, but these are the extinct ones, which we cannot work on at this moment. But there are certain objects we say like we want to convert the relationship between the rhinos, manatees, bats, and different rodents and elephants, for example, and the egg-laying mammals. So we want to, these are the objects, and we want to find the relationships between them based on certain traits. So the traits can be, so the objects can be species, as shown. They can be genes, they can be cell types, they can be different diseases, like how they're related. For example, how they're related. And for example, the different cancers, we know that the single cell genomics, so we can look at the cancer ancestral like evolution of cancers using the phylogenetic analysis. And some people also want to, like some sociologists, they're more interested in the how they're phylogenetic related using the languages, faiths, and the cards and the architectural styles, where they have the relationship on the basis of the faiths, cards, and the traits of the character. We classify them, or we characterize them, find the relationship based on certain features. For example, the features can be morphologic, how they look like, so molecular gene expression patterns. This can be the DNA sequences, or how they related, how the different organisms are related to the gene expression patterns. And also we can also look into the, like for example, the transcription factor binding sites. So we can look into the ancestral relationship to the phylogenetic relationship based on the transcription factor binding sites, motifs, and the domains as well. But the anthropologists, so they use the fossil, but there are some problems with using the fossils, because you might for the microorganisms, this is almost uncertain to find a fossil. and for number of organisms, the fossil information, we might find the incomplete fossil information. So, or it could be possible that the imprints and the timing of geological events. So the different continents are merged together and they're diverse. So it might be possible that the sum of the fossil might have been what you can say, they were diverse due to the geological events. So we might not get the exact information using the fossil records. So that's why today, or we'll say the currently, because of the advent of the next generation sequencing or the evolution of the sequencing technologies, we are more concerned about uh, we are more interested to use the phylogenetic tree based on the gene. So that's kind of paralog. The paralog is like the same gene, the gene duplication events. The homologous is the same gene in the same organism. Sorry, it's homologous in the same organism, and the same gene in the different organisms. So orthologs, or basically both of them are homologous. So one of the assumptions for the molecular phylogenetics is that the genes that homologs, that means they evolved from the same ancestor. So that's one of the assumptions we make when you create phylogenetic tree using the molecular sequence data sets. So we may not be able to cover all of them. So we'll cover most of them. So basic phylogenetics. We'll look into the characteristic traits and the different terminologies like the branches, lineages, topology, and how we can, from the alignment, how we can make the distance matrix different using the different models. Okay, are you having problem with the sound system? Can you hear me properly?
I can hear you, sir. Not sure about the rest. If they can mention in the chat.
So you do the alignment, and from that we create the distance matrices. We have seen how we can. There are different models you can use for the distance matrices. So here we'll cover like the Jukes and Cantor model. We'll just provide a highlight Jukes and Cantor model and the Kimura model. From distance, how we can create the trees. I think we might be able to call the PGMA and not deny for joining today because it might take longer. Then in the next class, we look into how we can create the trees using the different softwares. We'll not cover the Markov chain Monte Carlo trees of the Bayesian model. Okay. So one of the assumptions is like that. So if they came from the same ancestor, even we have some differences like yellow portions here, if they have the similar sequences, but although there are some minor differences, it means that they might have come from the same ancestor. For example, you find like there's a event, there is some mutations accumulated here, and again, this has been diverse from here, and it can be possible that you can have the same. So these two were diverse from these organisms, but all of them had the same ancestor. Yeah. So if you do the alignment and if you can find similar sequences, there is a possibility they might have the same ancestor. So, and from a common ancestor sequences to DNA sequences are diverse, for example, here, and each of these actually start accumulating the mutations. And that's the basis of evolutionary divergence. And this number of these mutations are used in molecular evolutionary analysis. So this is how you can calculate the molecular divergence using this accumulation of the nucleotides. For example, here, if the two sequences of length and differ from each other end sites, then the degree of divergence is small and divided by greater n. So let's have a look into here. So there's two sequences here. So we can find that the two mismatches. So the small n is my webcam is not working today. So yeah. Okay. So we found that the two differences, and the number of sequence, the length of the sequences are 1, 2, 3, 7. So, this is the divergence rate between these two sequences. This is how we find the degree of divergence between the two genes or the two sequences. So there is a strong relationship between the alignment and the first step before we do the phylogenetic analysis. We have seen that we use the different matrices and we do the alignment. And what does the alignment do actually? So multiple sequence alignment or the pairs alignment, they in the multiple sequence alignment we see that they sort of the sequences based on the similarity, and that is used as a guide tree for constructing a phylogenetic tree. And there are certain assumptions for the phylogenetic tree. So they assume that each position in a sequence can change independently from the other positions. That means they're not biased. And the gap, sometimes if you sometimes, for example, you put some gap here, yeah, to align the sequences. And what can happen, this gaps is actually, so this is a deletion for these sequences, and this is you can say it's an insertion. So if you know which sequence came before, so you can deduce which one is like an insertion, the deletion. So gaps in the alignments represent the mutations, insertion, deletion, and the genetics. So phylogenetic tree is an illustration of the evolutionary relationship among groups of organisms. We also call it dendrogram. And a tree is composed of the nodes, branches, and nodes represent the taxonomic units. Let's come here. So as we said that the trees can be inferred by the based on several criteria. For example, here like the skull of like the of different such, and you can see like there is the problem with using this criteria like the phenotypic characteristics is like, for example, you can create a phylogenetic based on the tree structure, so teeth structure, or for example the green shade here, like so this one is different from here actually, and this is absent in the other animals. So if you want to create a phylogenetic based on the green section, yeah, so you might find that some of them have been deleted. So it's very difficult to create a phylogenetic tree based on this feature. And the phenotypic feature is a lot less than the molecular sequences. And unless others they have a convergent evolution. So convergent evolution means like so for example, some organism has gained this feature again. So modern traits we'll look into one of the couple of next some of the next slides, the assumptions for the using the molecular data. So it's better to use the molecular data because you have a lot of features. And for example, if you look into this phylogenetic, the kangaroo elephant in the same group, and the dog, mouse, and the human in the same group, and it can be assumed that it's the difference between the ACA and the CCT. So that might be one of the couple of one of the features in the sequences that cause the divergence between these two groups. So we need to learn some terminologies here, the types of trees. So the trees, and for example, the root. There are two types of trees: rooted phylogenetic tree and the unrooted phylogenetic tree. Rooted is if we know which one is the ancestor, then we can create the root phylogenetic tree. That means like we can keep the ancestor at the baseline at the root, and we can see the divergence of the other sequences based against that root. But if you don't know which one is the ancestor, then we can't create a rooted phylogenetic tree. So in that case, actually, so what it does actually, it finds the software itself finds the divergence between sequences, and it takes most divergent two sequences, yeah, so, and finds which one is the middle. So then it makes the middle one as a root. So here the some terminology. So we look at the leaves. These are the outer branches. For example, these ones. These are the internal nodes. So that means sequence A and B diverge from the sequence 1, and the node 1 and 3 they diverge from the 2, and these are the branches. Sometimes these branches are like, we know the information about the branches, how the substitution or the time of divergent time, we know all this information. Sometimes we don't know. And the length of the branch represents the number of changes that occurred in the sequences prior to the next level of separation. For example, how many changes happened between these two before they are diverged from this node, internal node. But uh, yeah, so this is the external branches and the internal branches. We can like define them. For example, external branches are like F A F B G C G D, and the internal branches are H F H G I H. Yeah, so these are the internal branches. So in the each node, for example, these are the node F node, G node, they represent the speciation event, like when the two sequences diverge from each other, two species diverge from each other. So that's the event when they diverge from each other. And the branch connects two nodes of the tree. Usually, if I like the bifurcating trees, that means the two outer groups, two species diverging from the single node. Sometimes you find more than two. So CED, for example, you might find the 3 or 4 branches, sorry, 4 out groups here. So that happens when we have more than two species diverge from the single species or the single sequences, or we can't resolve actually which one came before which one's the ancestor, how the divergence happened. So that's also called the multifurcating or the radiation. Okay, so in a phylogenetic tree, if it's the amount of evolutionary time that passed from separation of the two sequences is not known, the phylogenetic analysis can only estimate the number of changes. For example, you compare the two sequences or three sequences, and you find out how many changes, amino acid or the nuclear changes among them. And after the branching event, one taxon can undergo more mutations than the other one. For example, from G to C, G to D. So it can happen that you might have the more mutation in this branch compared to this one. And the topology is actually the structure of the tree, is a branching pattern, how a tree is branched. So we might have to explain we found this topology, so this term more frequently. Terminal nodes, we also call them the outer taxonomic unit or the operational taxonomic units, or internal nodes we already discussed that represent the inferred ancestral unit. So these are the internal nodes, and this shows the ancestral unit. For example, the G is the ancestor for the C and D, and H is the ancestor for A, B, C, and D. So F is the ancestor. So where the two in this position, in this stage, that A and B diverge. So different kinds of trees can be used to depict the different aspects of it. For example, the cladogram, that's to simply show the relative recent common ancestor, how the, for example, how these descendants are related to common ancestors. Additive tree is a cladogram with branch length. Say, for example, like you can, from here you can say for red to this one, so two nucleotide changes happen. So from this red to here, so one nucleotide changes. From this two red, between these two reds, the three nucleotide changes. So you can see that the phylogram or the matrix trees, where there's the weighted trees. An alternative tree is a special kind of additive tree in which the tips of the trees are all equidistant from the root. So molecular clock hypothesis, that's another hypothesis, that's an assumption that all mutations occur in the same rate in all three branches. Yeah, and the rate of mutation is the same for all positions along the sequences. For example, if the A is the frequency of A in the column 1 is, for example, 0.2, this assumes that it will be 0.2 for the all positions. Like if you look at number 7 position, 120 positions, the frequency of each sequences will be the same. So that's the assumption here. And molecular clock hypothesis is most suitable for the closely related species. And using the molecular hypothesis, you can find out the divergence time of between different sequences. Yeah. Say for example, here we see the time of divergence in millions of years, and the cytochrome C residue in different organisms. For example, like around 250 years, the millions of birds versus a reptile. So the divergence time between the birds versus, so on the x-axis you put the time divergence, because this is known from the fossil records, so how many years ago they diverge from each other. So if you put the amino acid substitution on the y-axis, and you can determine the time in how many years ago. For example, if you like say that the amino acid substitutions between the two organisms is 35, yeah. So if you go here, so they diverge around 800 million years ago, or the 780 million years ago. So that's from this graph you can determine the based on the amino. So you're given two sequences or the three sequences, and if you know the divergence time, you can find from this standard curve the how many millions years ago they diverge from each other. So that's a good thing about the molecular clock hypothesis. So as I said, there are two kinds of trees can happen. If you know the ancestral information, we can draw the cladogram. So you have the root. So you put the ancestor in the root, and you can see that how the other sequences diverge from each other. So, and the path from the root to the nodes corresponding to their evolutionary time. For example, from this root to the different nodes, it shows the evolutionary time. So the time divergence, yeah. And the unrooted tree, that's the phenogram, where all objects are interrelated. So you depends it's only based on the how the changes on the nucleotides of the amino acid, how many changes happen among the organisms, among the sequences. So based on that, you can create the phenogram. So you don't have the information about the ancestral information. You just know the substitution rate or the changes in amino acid, the nucleotides among them. So as I said that we can create the phylogenetic tree based on the physiological traits and using the molecular traits. So traditional phylogenetics, you can use the species tree based on the different phenotypic characteristics and small number of traits for the hooves, snails, teeth, and the horns. You can so see we having some organism having the same horns, they can classify the similar group, and we will be traits such as the arrows one, the parsimony. We'll look at that later on. So parsimony means like the minimum number of traits you require to classify the organism or the differentiate the organisms. So the modern phylogenetics build on the gene trees and the species trees, and that means that the phylogenetics can be based on a single gene, or it can be based on the species information of very large number of traits. For example, nowadays you can do the whole genome sequences, and you look into every DNA base and every protein residues. If you are aligning the amino acid sequences, these methods use every residue and finds a best tree for you. But I'll come to that. So back mutations are very problematic, and a small number of letters arise many times independently. Let's come to so this is the common phylogenetic tree terminologies, as I said. So you have the root, and this is the branches. So and this is the internal nodes. We can never get back to this nodes. Actually, these are like based on the algorithm of the calculations. But we can work on this one, the outer these taxes like the A, B, C, and D, the outer taxonomic units, operational taxonomic units. These internal nodes actually the divergent point when the sequences diverge from each other. So why we do the molecular phylogeny? There are certain advantages. So they're more numerous than the fossil records and easier to obtain. Yeah. So as I said, it's difficult to obtain the fossil records, and there's no sampling bias involved. For example, you're taking the molecule. For example, you're getting the what you can say the fossil from the one location for the same organism, and you're taking the fossil record from the same organism from the different places. Yeah. So it could be possible the sampling bias, and sampling bias can what can you might have asked with the phylogenetic tree, so that might give the clear picture of the phylogenetic relationship or the ancestral relationships. You get them using the molecular sequence data. You can get the more clear cut in the robust phylogenetic trees compared to the other features. So this can be defined as the study of evolutionary relationships of genes and other biological macromolecules by analyzing mutations. So you look in the mutations at the different positions in their sequences and develop the hypothesis based on the changes of nucleotides or the amino acids in the different positions. And you can create the evolutionary relatedness based on this molecular sequence data. And it has certain assumptions. For example, they are homologous. That means they came from same ancestors. So when you're using the phylogenetic tree, when you're using the molecular sequences data for the phylogenetic tree constructions, one of the assumptions I make is that they came from the same ancestor. And another one, it is assumed that it's bifurcating. Yeah, bifurcating means, for example, you have a node here. So you get the, usually you get the two species or the two genes, two events that diverging from the same sequences. Yeah. So it's a bifurcating, meaning that the parents present two daughter branches at a given point. And each position in a sequence evolve independently. As we said, therefore you look for the different positions, 3, 4, whatever it is, and they are all evolved independently. And some positions you'll find they're like conserved, and some position you'll find they're diverse from each other, so they're different from each other. So, and the variability, the differences in the sequences are sufficient informative. They'll give the sufficient information for constructing the unambiguous phylogenetic tree. For example, it might be happen that, as you said, like you take sequences and they're more than 90% identical, so you don't get enough information there to create a phylogenetic tree. And you take, for example, another way, like you take few sequences and some of them are only 10 or 15% similar to each other. So you don't get enough information because they're highly diverse. So they should be homologous. And as I said, that the parent is two daughter, and these are the assumptions you need to make before you create a phylogenetic tree. So there, we already discussed this one in one of our, so simple cladogram. So it's just show the topology, how the different organisms, like the different taxons, are different from each other. Just a simple cladogram. And here you have the chronogram. You provide the time, for example. So just a time 1, time 2, and the between the time the diversification between these two groups. So this attack this group and this group, the divergence is between the T2, this. So this is the position when they diverge from each other. Yeah. So you have simple cladogram, and if you put the time divergence time, that becomes a chronogram, topology divergence time. And what you can, so you can show the divergence rate actually. So for example, that taxon B substitution, there are 6 changes happen here. So 1 changes happen here, and for the taxon B 5 from the root, the 5 changes happen here, and the 3 changes happen here, and after that they diverge into 2 groups. And this is, so you have the topology, divergence time, and the divergence rate. So gene phylogeny versus the species phylogeny. Okay. Okay. So say for example, as we said that at the beginning, when the scientists started to work on the phylogenetic tree, because of the sequencing facilities, they used only single genes, yeah. But the problem is that, so the single gene will not give you the correlate with the evolutionary path of the species. For example, the reason is that, so it might be possible for you have like many genes, yeah. So it could be possible that this is more variable compared to this one. Yeah, so this is less variable, yeah. So if you create a tree with the more variable genes, you'll find a tree that is different from the tree which you create with the less divergent sequences. So using the single sequence is not a good idea at all. So species evolution, that's what it's saying, that is a combined result of evolution by multiple genes. For example, you have an organism here, you have an organism here, and the A, B, and C. So it has many genes, for example, here. And say for example, you also have many genes here. So if you just take on a single genes, so it will not give you the proper. So what you can do, so what we do actually, we take a number of genes. For example, we take the genes that are less variable among them. For example, I can take 10 genes from here, and the similar 10 genes orthologs from here, and the same 10 genes from here. Yeah, and you combine these 10 genes and make a one gene. Yeah, they do the same thing for this. So you take the ortholog and make it. So then you create a phylogenetic tree based on the similar genes, but we're using the multiple genes together. So there is one very important studies before the next generation sequencing evolved is a multi-locus sequence typing. Okay, what is it? What so for the same species, for example, it take a number of housekeeping genes are concatenated and phylogenetic tree is inferred. For example, you take the usually 7 to 10 genes are taken. Yeah. So and based on the, for example, you take the 1, 2 gene 3, gene 4, all the series of 4, 5, yeah, and they categorize based on, for example, if it's for the gene 1, yeah, if a certain sequences, you get the type, for example, 7, yeah, and for the gene 2, if you find certain sequences, you give a type of 5 based on the gene sequences. Yeah. So for the 3, you give, for example, the type 4. For the sequence number 4 outside, yeah, for sequence number 4, you give 3, yeah. For the sequence number 5, based on the sequences, you said this is a type 4 again, yeah, something like that. Okay, this is for the one organism or the one species. For the E. coli, yeah, E. coli strain 1 or something like that. If for another E. coli in your lab, so you said that found this based on the sequences, this is type 6, 4, 4, yeah. So you find the different pattern for this one, yeah. And you take another E. coli, and it said that okay, so 4, 3, 4. So if you find this pattern, you say this is a sequence type something, yeah, sequence type X. If you find this pattern, you say this is a multi-locus sequence type Y. Yeah, and if you find this type, because this one is this similar, so you said this is also sequence type X. So based on the multiple genomic regions, you can create a phylogenetic tree and the sequence type similarity or the dissimilarity relationship among the organisms based on the several housekeeping genes. For creating a phylogenetic tree, you need to provide the information to the computer, and one of the file format is called a Newick format. For example, that can be weighted or the unweighted. For example, if the distance is not known, yeah, so you can put like B and C together, and B and C makes branches with the A. So these three together. So sorry, my webcam, as I said, the webcam is not working. So it's, and the D and E is another branch. So they make another branch. But here, if you look at that, so this is weighted. So you know the distances here, evolutionary even. For example, from the here, you can't. So this is, if this is 1, the length is 2 here, the length is 1 here, and the length is 2 here. Yeah. So that means B is 1, C is 2, comma, and from here A is 2. Yeah. And here, like this is 1.2, and this is double of the D. So that means the D, another branch, 1.2 and the 2.5. This is the Newick format. So this is just, I just had to show you. So that's how the different file formats work. One of the format is a Newick format. This is used for phylogenetic tree construction by the computer programs. So don't need to worry about this format. Okay. Now, creating tree is very difficult. Yeah. The reason is that, so whenever we give sequences, for example, if you give like thousands of sequences, so what the software does based on the different algorithms or the algorithms, it creates a lot using the, for example, if you get the few sequences, they'll create a number of trees based on the number of sequences you provide, and it will choose the best tree for you. Okay, and say for example, there are 3 trees. So A, B, C. You see here, there's a one pattern here, E and F. A, B, C, D is always branching together. Here, A, B, C, B is branching together. So, and out of 3, twice the 2 times A, B, C, and D comes together. So the computer will assume that this is the best tree. So it will put A, B, C, and D together, and E and F together. Yeah. So this is the how the software does creates it. So it creates a lot of trees and finds a consensus tree for the analyst. The formula for the rooted tree is 2n minus 3 factorial 2n minus 3 to the power. So say for example, if we give the 5 sequences or the 5 sequence of the 5 organisms, then you can get the 100. It creates 100 of 20 trees and gets the best out of 120 trees. It gives us the only one best tree. And if it's for unrooted tree, okay, so you need to calculate using this formula. And for example, here's for the, if you have 6 taxons, yeah, so it will get 105 unrooted trees and 945 rooted trees. So getting the best tree is very difficult because they have to do so many calculations. So with the 4 taxa, you can get. So this is like typical number of taxa. So as the number of taxa increases, number of tree topologies also increases. If you look into this, if you have like around 20 taxons or the 20 sequences, it creates around 10 to the power 18 unrooted tree and 10 to the power 20 rooted tree. So this is the number of trees it creates, and then it gives us the best tree out, best consensus tree out of this number of sequences. So we already discussed about in details in the multiple sequence alignment which one you should use. Obviously, if it's coding sequences, we'll go for the amino acid sequences to create the phylogenetic trees. So, and if it's a closely related organism, nucleotide sequences which evolve more rapidly than proteins can be used. And so, for example, the evolutionary analysis of different individuals within a population, non-coding regions of mitochondrial DNA are often used. And as I said, the Richard III, the one of the king that died, and his dead body was found near the cathedral. It was not in the cathedral, near a garage in a cathedral in Leicester. So to confirm that that's the dead body of the Richard III, the sequences mitochondrial DNA of Richard III and compared that one couple of the descendants, one living in UK and another living in the Canada, and then compared the mitochondrial non-coding DNA to confirm the family relationships. And if you're studying the evolution of more widely divergent group of organisms, so we use the ribosomal RNA sequences or the protein sequences. You know, the 16S for bacteria, we use the 16S ribosomal RNA sequences because they're more conserved, so very little changes occur in the ribosomal RNA. But if you want to like determine the relatedness in the deepest level, for example, between bacteria and the eukaryotic, you can use the homologous genes or the orthologs between the genes, orthologs between the bacteria and the eukaryota, using the conserved protein sequences, and it makes more sense than using the nucleotide sequences. Okay, and we know there are certain reasons for using the protein sequences because so one protein sequence, the amino acid sequence, can be coded by the different nucleotide sequences. So if you compare the nucleotide sequences, you might find that they're different from each other, but it could be possible that the changes is a synonymous changes. It's not the non-synonymous changes. So synonymous changes mean the changes of nucleotide, if that changes in the amino acid, that we call the non-synonymous changes, and if the changes in the nucleotides correspond with the amino acid changes, we call the non-synonymous substitutions or the changes. So meaning that the changes in the codon may not result in changes in amino acid. The protein sequences can remain the same while the corresponding DNA sequence have more room for variation, especially at the third codon position. So we know that because they're due to the changes in that's the degenerate region of the codons. Okay. The significant differences in the evolutionary rates among the three nucleotide positions also validates one of the assumptions rebuilding. In contrast, the protein sequences do not suffer from this problem, even for divergent sequences. So DNA sequences are sometimes more biased than the protein sequences because of the preferential codon uses in the different organisms. For example, for a single amino acid, one organism may use the one codon, whereas the other organism might use a different codon for the same amino acid. So there is a bias, codon bias for certain organism for certain amino acid. And in this case, a different codon for the same amino acid reuse at the different frequencies. And that will lead to the sequence variation, that is not basically attributed to the evolution. What it means actually, so even though you may find that, okay, the nuclear changes happen, and you might think that, okay, it might be due to the evolutionary changes, actually, which is actually not. Because there hasn't been any changes at the amino acid level. So, okay. In addition, the genetic code of mitochondrial varies from the standard genetic code. So let me show you something. So, genetic code table. So you can see this is standard table. So there are different tables. If you go to the NCBI, you'll find the different tables for the different organisms. So the codon might vary from this is standard code table. So A is standard start codon, and for the vertebrate mitochondrial code, so there is another table that's called the table 2. So you have the different from the standard code. For example, AGA codes for the termination code in the code 2, and it's standard actually it's arginine. But in case of the vertebrate mitochondrial, so an alternative initiation code for the Bosteros is AUA. So we have the table for the yeast mitochondrial code. So in the code 3 is AUA codes for the methionine, CUA codes for the threonine, whereas it goes to the leucine, and standard code is for the leucine. So like that, you have 33 tables. So that's what it's saying. So in addition, the genetic code of mitochondria varies from the standard genetic code. Therefore, comparison of mitochondrial protein coding genes, it is necessary to translate the DNA sequence and the protein coding sequences. So if you compare the nucleotide sequences, so you'll find that there are differences. Actually not. At the amino acid level, there might not be changes because they use a different standard genetic code table. And it has been shown that the randomly selected the DNA sequences can result up to 50% sequence identity, whereas the gaps are allowed compared to only 10% at the protein sequence level. And when you compare the protein coding DNA sequences, if you allow the gaps, yeah, so there almost always cause the frameshift mutations, errors, making the alignment biologically meaningless. And protein sequences clearly have a higher signal to noise ratio. Protein-based phylogeny in most cases may be more appropriate than the DNA-based phylogeny. So if you know that your sequence is a coding sequence, it's better to translate that and translate that using the stand genetic code table, so specific for that organism. So sorry for this one, I just copied it from directly from the book. So DNA sequences can still be very informative some cases. For example, those closely related organisms, DNA sequences depict the synonymous and the non-synonymous. We know that if the changes in the nucleotide sequences that result in the amino acid changes, we call them the non-synonymous. And if the changes in the DNA nucleotides doesn't correspond to the, doesn't result in the amino acid changes, that's called the synonymous substitutions or the changes. So there is certain advantage of the synonymous and the non-synonymous substitutions. Yeah. So if your synonymous substitutions is greater than this, so usually what organisms tend to like, actually, they don't like changes. Yeah. So they don't like changes in the amino acid. Usually it's very rare. But if, for example, for survival, a good this can happen. For example, you're comparing two sequences, yeah, and you found that the in one sequence in just write it down. So you align the two sequences, and you found that the A and C in one sequence is in certain positions, that's A, and now the sequence is C. Yeah, and it could happen that you find the, like, comparing the two sequences and you found G and G. Yeah. But they might not be the true reflections of what actually happened in real life. It could be possible that this A to C, actually, what are you seeing today, actually, there are multiple events happen before this changes happened. For example, A might have gone to T, and the thymine might have been changed to guanine, and that ultimately came to C. But when you compare the two sequences, you can see only the A changed to C. So we are missing, actually. So we are missing these two events from our, so that can mislead the phylogenetic analysis. And it can, as we said, that there might be possible that this G and G, there might be some changes happening in the middle. For example, G changes to C and C changes to G. Yeah, but we can see only the G to G. There we can, the software might think that the only G changes to G. There are no changes, actually, but in real life, G changes to C in the middle, and that ultimately mutated to the guanine. So this kind of misleading information is called the homoplasy. So there we need some algorithms. So if they are not corrected, that might lead to the generation of the incorrect trace. So to correct the homoplasy, statistical models are needed. It's still they are not developed properly. So some of you might, if you're interested, you can work on that to infer the true evolution distance between the sequences. So we covered this one, the characters, the basics of the phylogeny, the traits, branches, lineages, topology, and we didn't cover this one. So I'll just go through. So how do we create the phylogenetic tree? So from the nucleotides of the peptide, so you do the multiple sequence alignment. And from that, with the distance matrix, and we create the phylogenetic tree. And this is evolutionary history represented as a binary tree. So there are two basic approaches we follow. One, the distance-based method we'll discuss in the next class. And another one is the character-based method. So distance-based method is we create a matrix as we did for the Needleman and Ohms and these. So from that distance-based method, from that distance matrix, we can create the phylogenetic tree. We get something like that, yeah. And character-based method, we look into every character, each individual character means each and every column, nucleotide of the amino acid sequences. Then from that we create the phylogenetic tree. So this is more robust, and but for if you have the many sequences, it's very time consuming. So in that case, we prefer the distance-based method. Okay. So before we create the distance-based method, we need to choose a model actually, yeah. So two most favorite model is like Jukes and Cantor model. So Jukes and Cantor model, what it assumes that the changes in the every site of the sequences as if all nucleotides are substituted with equal probability. For example, the changes between the A to T and the A to C, A to G has the equal probability. That's 0.25%. And this is a formula you use. For example, the distance of two sequences minus 3 by 4 ln 1 minus 4 by 3, the probability of changes between the two sequences. Yeah. Observed sequence distance measured by the proportion of substitution. For example, you have two sequences and length of 10. So the nucleotide changes we found, for example, the 3. So 3 changes out of 10 sequences. That means the probability of distance is 0.3. So if you have that value, for example, 0.3, if you put that, for example, if an alignment of sequences A and B is 20 nucleotides long and 6 pairs are found to be different, that means 6 divided by 20 is 30% divergence. So observed distance PAB is 0.3. So if you put that value, so distance between two A and B is 0.38. So this, you have to understand this one, because in the next class we look at how from this distance matrix will create the phylogenetic tree. So if you're given the A and B, so if you're given hundreds of sequences, so using the Jukes and Cantor model, using this formula, you can create a distance, the matrix table for the difference, the distance between the A and B, A and C, A and D, and the different taxa using this formula. So for example, if you have like 4 nucleotide changes, so you can say the 4 divided by 20, that means 50% changes, yeah, 0.5. So this is how you do that. And but in real life, it's very unusual that for example, this is like Jukes and Cantor model. They say that okay, the changes between the T, A and A, there will be no assumptions, but in T and A, you have the same rate alpha, alpha, alpha. That means it's at the same rate, the 0.25 actually. So the substitution rate between T and A is alpha, G and A is alpha in that sequence. That's the Jukes and Cantor model. But Kimura came up the model that in real life it doesn't happen actually, yeah. So transition is more frequently happen than the transversion. Transition means like from A to G or T to C, there's a transition, or A to T on A to C, or G to T, or A to, yeah. So that's the transversion. So transition is more frequent than the transversion, and using that, he developed this formula. So he looked into that PTI means the rate of transition. So you're given the two sequences, and you find the rate of transition changes transition and rate of transversion. And if you have the transition and the transversion rate, you can find the distance using this Kimura model. For example, so they differ by 30% if 20% are result transition and 10% changes are result transversion. That means that 20.2 is transition and 0.1 is transversion between the A and B two sequences. So the distance between them is 0.4. So I'll not go into the corrections, how you correct this one. So this is, then you can create the transition and using the Kimura model, the distance between the A and C, B and C, D, A and C, and Using that, you can create a distance matrix table, and that distance matrix tables can be used for creating the phylogenetic tree. So as we said, so the between T and A is different compared to the A and G, because A and G is more frequent than the changes between the A and C and the A and T. So transition is more frequent than the translation. So Kimura used this model. So I'll not go into the details. So I'll finish it here. So if you have any questions, I'll upload the lecture. Sorry. Okay. So thanks. Let me go through the questions. I think Mr. Mohsin Mannan, we already discussed this one. Yeah, so it is possible that the variation of the same residue point mutation during the evolutionary window period may revert back to the residue that was at the position primarily. Yeah, so that we call the homoplus, and still there is a room for improvement of algorithm to address this issue. Would we know whether or not the residue underwent any changes? Yeah, so that's the challenge. That's the biggest challenge, Mr. Mohsin. Yeah. Last assignment will upload this week. Okay. That's exactly what I asked. Couldn't we track the changes that may? Yeah. So until the last few years, if you're not working with the same organism, if you're not with specific organism, it's very difficult to say actually. So that's why we call it the home plastic. That's a very challenging area. Yes. So any more questions? Yeah, you're welcome. So in the next session, I'll try to make it easy. I'll just not go into the details of the algorithms. Next class, well, this will be more hands-on. We'll go through the Mega software most probably and the CVU, how to create the tree. And I'll also introduce the fig tree. So that's very useful to make nice trees for your publications. Yeah. Wakas, do you want to say something?
Yeah, so thanks, Maksud. I just wrote two announcements in the Zoom chat box. Number one is that we will upload our last assignment by the end of this week. Second, there will be no Zoom session next week. So the next Zoom session will be on 24th of August. And so if Maksud is interested and all the participants are interested as well. So earlier the schedule which we draw, it's so we have no lecture on 17 August, means next week. But if Maksud, you can have your the continuation of the same next lecture if you are interested. As you mentioned that you can have some on the you can some hands-on on the phylogenetic tree or its analysis of fig view, etc. So you can go for 17 August, but for the scheduled stuff, the next Zoom session will be on 24th of August. So Maksud, can you take on 17 August lecture?
I think I planned according to 24th because what you planned previously. So okay, I like postponed a number of assignments on that day. So it's
because for this phylogeny we have only one lecture which you just delivered, and then we have a gap on 17th of August, and then we have 24th of August. I will start for the structural bioinformatics on the 24th of August. But it's up to you if you are.
Okay, so you had only one lecture, so I thought that I have another lecture on the 24th. So yeah, then it's fine. I can have 17th for the hands-on. Yeah, I got it. Yeah.
Okay, so that's great. So if anybody is interested in a phylogenetic tree and analysis, so Maksud will take the next Zoom session on 17th of August. Then I will continue on 24th of August from the topic will be the structural bioinformatics, protein structure and function relationships. Okay, so that's I think I just want to say. So if there's any other query or anything, just let us know. Or Google Classroom is always there to discuss the assignment, to discuss the any topic or query. Yeah. Yeah. So if there is no more queries, so I think we can sign off, Maksud. Okay.
Okay, yeah. Thank you very much. Thank you, everybody.
Thank you. Thank you very much. See you next week. Okay. Bye bye. Take care.